welcome back to my YouTube channel. I am Rajshekhar from the Department of CSC Cybersecurity from the SVC College. Today we are discussing about the subject Biology for Engineers. The topic is describe the nucleic acids and properties of nucleic acids. About the topic is nucleic acids are biological macromolecules that play fundamental roles in the storage, transmission, and expression of genetic information in living organisms. They come into the two main types, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. Deoxyribonucleic acid DNA is a typically double-stranded and forms a double helix structure composed of two polynucleotide chains. Components each strand consists of nucleotides containing a phosphate group, a deoxyribose sugar molecule, and one of four nitrogenous bases adenine, cytosine, guanine, and thymine. Function: DNA stores genetic information in the sequence of its nucleotide bases. This information is crucial for protein synthesis, cellular function, and inheritance. RNA ribonucleic acid structure is RNA is a generally single-stranded, although it can form complex secondary structures through base pairing. Components is like DNA, RNA is composed of nucleotides, but it contains ribose sugar instead of deoxyribose, and uracil replaces thymine as a base pairing partner of adenine. Function: RNA plays diverse roles in gene expression, including mRNA, messenger RNA, which carries genetic information from DNA to the ribosome from protein synthesis. tRNA transfer RNA, which helps in translating mRNA into proteins, and various regulatory RNAs that control gene expression. Introduction to nucleic acids. Nucleic acids are essential biomolecules that carry genetic information within cells and are responsible for transmitting hereditary traits. These complex macromolecules are found in all living organisms and play a crucial role in cellular processes, including protein synthesis, growth, and development. Structure of DNA and RNA. DNA, deoxyribonucleic acid, is a double helical structure and composed of two antiparallel polynucleotide chains. Each chain consists of nucleotides linked together by phosphodiester bonds. These nucleotides are made up of a deoxyribose sugar, a phosphate group, and one of four nitrogenous bases adenine, guanine, cytosine, and thymine. RNA is a single-stranded nucleic acid that plays a crucial role in protein synthesis. It consists of nucleotides composed of a sugar, ribose sugar, a phosphate group, and one of four nitrogenous bases adenine A, guanine G, and cytosine C and uracil U. RNA comes in various forms, including messenger RNA and transfer RNA, tRNA, and ribosomal RNA. Nitrogenous bases in nucleic acid are adenine. Adenine is a purine base that pairs with thymine T in DNA and uracil in RNA. Guanine is another purine base that pairs with cytosine in both DNA and RNA. Cytosine is a pyrimidine base that pairs with guanine in both DNA and RNA. Thymine. Thymine is a pyrimidine base that pairs with adenine in DNA only. Uracil is a pyrimidine base that pairs with adenine in RNA only. Phosphate and sugar backbone. A sugar in DNA is deoxyribose, while the sugar in RNA is ribose. These sugars form the backbone of the nucleic acid chains. Phosphate groups link the sugars together to form the polynucleotide chains. These phosphate groups give nucleic acids their acidic properties. Nitrogenous bases. The nitrogenous bases are attached to the sugars and project outward from the backbone. They are responsible for the genetic code and the pairing of the DNA strands. Double helical structure of DNA. Antiparallel strands. The two strands of DNA run in opposite directions, anti-parallel, and are held together by the hydrogen bonds between the nitrogenous bases. Base pairing: adenine A always pairs with thymine and guanine always pairs with cytosine. This specific pairing is called complementary base pairing. Major and minor grooves: the double helix has two grooves. A major groove and a minor groove provides access for proteins to bind the DNA molecule and they read the helical structure. Replication of DNA. Unwinding: the double helix unwinds by enzymes called helicases and which breaks the hydrogen bonds between the base pairs. Priming: short RNA primers are synthesized by an enzyme called primase. These primers provide a starting point for DNA polymerase to add nucleotides. Elongation: DNA polymerase adds nucleotides to the new strand in a 5 to 3 direction following the base pairings rules. Termination: the process terminates when the entire DNA molecules is replicated, resulting in two identical DNA molecules. Trans- Transcription of DNA to RNA. DNA double-stranded deoxyribo at the higher thymine and located in the nucleus. RNA is a single-stranded ribosugar and uracil found in the nucleus and cytoplasm. Translation of RNA to proteins. Ribosomes. Ribosomes are the sites of protein synthesis. They read the mRNA sequence and bring together amino acids to form a polypeptide chain. Amino acids. Amino acids are the building blocks of proteins. They are brought to the ribosome by tRNA molecules which contain anticodons that match the codons on the mRNA. tRNA transfer RNA is the molecules transfer amino acids to the ribosome. They have a specific anticodon that matches a codon on the mRNA sequence. Properties of nucleic acids: hydrophilic, acidic, and long chains. In the next slide, we can deeply discussing about the properties. The properties of nucleic acids are the polymer structure. Nucleic acids are polymers composed of nucleotide monomers linked together through phosphodiester and bonds. Each nucleotide consists of a phosphate group, a 5-carbon sugar deoxyribose in DNA and ribose in RNA, and a nitrogenous base adenine, guanine, cytosine, and thymine in DNA. Adenine, guanine, cytosine, and uracil in RNA. Sequence specificity: the sequence of nucleotide bases along the nucleic and chain determines its genetic information content. This sequence encodes instructions for protein synthesis and other cellular functions, making it crucial for inheritance and cellular activity. Complementary base pairing: in DNA, adenine pairs with thymine and guanine pairs with cytosine through hydrogen bonding. In RNA, uracil replaces thymine. This complementary base pairing allows for precise replication of genetic information and facilitates processes like transcription and translation. Double helix structure: DNA molecules typically exist as double helices where two polynucleotide strands wind around each other in a spiral. This structure provides stability and protection to the genetic material, with the bases stacked inside the helix and a sugar phosphate backbone forming the outer structural support. Flexibility and conformational dynamics, genetic stability and functional diversity of RNA, change, charge and solubility, molecular recognition. These are the properties of the nucleic acids. Importance of nucleic acids in biological processes: genetic information, protein synthesis, cellular regulation, and evolution. These are the nucleic acids biological processes. The conclusion is nucleic acids are the molecular cornerstone of life, essential for storing, transmitting, and expressing genetic information in all living organisms. Comprising DNA and RNA, nucleic acids exhibit remarkable diversity and functionality, enabling the intricate processes of heredity, protein synthesis, and cellular regulation. Their structure, characterized by a sequence of nucleotide bases adenine, cytosine, guanine, and thymine or uracil forms the basis of genetic coding and molecular recognition. Through complementary base pairing, nucleic acids facilitate the faithful faithful replication of genetic material and synthesis of proteins, thereby ensuring the continuity and functionality of living systems. Thank you guys. And thanks for watching the video guys.